Acetylcholine.
Acetylcholine is an organic molecule that acts as a neurotransmitter in many organisms, including humans. It is an ester of acetic acid and choline, with chemical formula CH, 3COO, CH, 2, 2N+, CH, 3, 3 and systematic name 2-astoxy N N-trimethylethanaminium. Acetylcholine is one of many neurotransmitters in the autonomic nervous system, ANS. It acts on both the peripheral nervous system, PNS, and central nervous system, CNS, and is the only neurotransmitter used in the motor division of the somatic nervous system. Acetylcholine is also the principal neurotransmitter in all autonomic ganglia. In cardiac tissue, acetylcholine neurotransmission has an inhibitory effect, which lowers heart rate. However, acetylcholine also behaves as an excitatory neurotransmitter at neuromuscular junctions in skeletal muscle.
History.
Acetylcholine, ACH, was first identified in 1915 by Henry Hallett Dale for its actions on heart tissue. It was confirmed as a neurotransmitter by Otto Loewi, who initially gave it the name vagustov because it was released from the vagus nerve. Both received the 1936 Nobel Prize in Physiology or Medicine for their work. Acetylcholine was also the first neurotransmitter to be identified.
Function.
Acetylcholine has functions both in the peripheral nervous system, PNS, and in the central nervous system, CNS, as a neuromodulator. Its receptors have very high binding constants. In the peripheral nervous system, acetylcholine activates muscles, and is a major neurotransmitter in the autonomic nervous system. In the central nervous system, acetylcholine and the associated neurons form a neurotransmitter system, the cholinergic system, which tends to cause anti-excitatory actions.
In the peripheral nervous system.
In the peripheral nervous system, acetylcholine activates skeletal muscles, and is a major neurotransmitter in the autonomic nervous system. Acetylcholine binds to acetylcholine receptors on skeletal muscle fibers. It opens ligand-gated sodium channels in the cell membrane. Sodium ions then enter the muscle cell, initiating a sequence of steps that finally produce muscle contraction. Although acetylcholine induces contraction of skeletal muscle, it acts via a different type of receptor, muscarinic, to inhibit contraction of cardiac muscle fibers. In the autonomic nervous system, acetylcholine is released in the following sites. All pre- and postganglionic parasympathetic neurons, all preganglionic sympathetic neurons. Ganglionic sympathetic fibers to suprarenal medulla, the modified sympathetic ganglion. On stimulation by acetylcholine, the suprarenal medulla releases epinephrine and norepinephrine. Ganglionic sympathetic fibers to suprarenal medulla, the modified sympathetic ganglion. On stimulation by acetylcholine, the suprarenal medulla releases epinephrine and norepinephrine, some postganglionic sympathetic fibers. Pseudomotor neurons to sweat glands, pseudomotor neurons to sweat glands.
In the central nervous system.
In the central nervous system, ACH has a variety of effects as a neuromodulator upon plasticity, arousal and reward. ACH has an important role in the enhancement of sensory perceptions when we wake up and in sustaining attention. Damage to the cholinergic, acetylcholine-producing, system in the brain has been shown to be plausibly associated with the memory deficits associated with Alzheimer's disease. ACH has also been shown to promote REM sleep. Recently, it has been suggested that acetylcholine disruption may be a primary cause of depression.
Pathways.
There are 3 ACH pathways in the CNS. Pons to thalamus and cortex, magnocellular forebrain nucleus to cortex, SP to hippocampal.
Structure.
Acetylcholine is a polyatomic cation. It and the associated neurons form a neurotransmitter system, the cholinergic system from the brainstem and basal forebrain that projects axons to many areas of the brain. In the brainstem it originates from the pedunculopontine nucleus and laterodorsal tegmental nucleus, collectively known as the mesopontine tegmentum area or pontomesencephal tegmental complex. In the basal forebrain, it originates from the basal optic nucleus of minute and medial septal nucleus. The pontomesencephal tegmental complex acts mainly on M1 receptors in the brainstem, deep cerebellar nuclei, pontine nuclei, locus caeruleus, raphe nucleus, lateral reticular nucleus and inferior olive. It also projects to the thalamus, tectum, basal ganglia and basal forebrain. Basal optic nucleus of minute acts mainly on M1 receptors in the neocortex. Medial septal nucleus acts mainly on M1 receptors in the hippocampus and neocortex. In addition, ACH acts as an important internal transmitter in the striatum, which is part of the basal ganglia. It is released by cholinergic interneurons. In humans, non-human primates and rodents, these interneurons respond to salient environmental stimuli with stereotyped responses that are temporally aligned with the responses of dopaminergic neurons of the substantia nigra.
Excitability and inhibition.
Acetylcholine also has other effects on neurons. One effect is to cause a slow depolarization by blocking a tonically active K+ current, which increases neuronal excitability. In alternative fashion, acetylcholine can activate non-specific cation conductances to directly excite neurons. An effect upon postsynaptic M4 muscarinic ACH receptors is to open inward rectifier potassium ion channel, CuR, and cause inhibition. The influence of acetylcholine on specific neuron types can be dependent upon the duration of cholinergic stimulation. For instance, transient exposure to acetylcholine, up to several seconds, can inhibit cortical pyramidal neurons via M1-type muscarinic receptors that are linked to GQ-type G protein alpha subunits. M1 receptor activation can induce calcium release from intracellular stores, which then activate a calcium-activated potassium conductance, which inhibits pyramidal neuron firing. On the other hand, tonic M1 receptor activation is strongly excitatory. Thus, ACH acting at one type of receptor can have multiple effects on the same postsynaptic neuron, depending on the duration of receptor activation. Recent experiments in behaving animals have demonstrated that cortical neurons indeed experience both transient and persistent changes in local acetylcholine levels during cue detection behaviors. In the cerebral cortex, tonic ACH inhibits LAIR4 medium spiny neurons, the main targets of thalamocortical inputs, while exciting pyramidal cells in LAIRs 2/3 and LAIR5. This filters out weak sensory inputs in LAIR4 and amplifies inputs that reach the LAIRs 2/3 and LAIR-L5 excitatory microcircuits. As a result, these less specific effects of ACH might function to improve the signal-noise ratio of cortical processing. At the same time, acetylcholine acts through nicotinic receptors to excite certain groups of inhibitory interneurons in the cortex, which further dampen down cortical activity.
Decision-making.
One well-supported function of acetylcholine, ACH, in cortex is increased responsiveness to sensory stimuli, a form of attention. Phasic increases of ACH during visual, auditory and somatosensory stimulus presentations have been found to increase the firing rate of neurons in the corresponding primary sensory cortices. When cholinergic neurons in the basal forebrain are lesioned, animals' ability to detect visual signals was robustly and persistently impaired. In that same study, animals' ability to correctly reject non-target trials was not impaired, further supporting the interpretation that phasic ACH facilitates responsiveness to stimuli. Looking at ACH's effect on thalamocortical connections, a known pathway of sensory information, in vitro application of cholinergic agonist carbacetyl to mouse auditory cortex enhanced thalamocortical activity. In addition, Gill et al. 1997, applied a different cholinergic agonist, nicotine, and found that activity was enhanced at thalamocortical synapses. This finding provides further evidence for a facilitative role of ACH in transmission of sensory information from the thalamus to selective regions of cortex. An additional suggested function of ACH in cortex is suppression of intracortical information transmission. Gill al. 1997, applied the cholinergic agonist muscarine to neocortical layers and found that excitatory postsynaptic potentials between intracortical synapses were depressed. In vitro application of cholinergic agonist carbatyl to mouse auditory cortex suppressed intracortical activity as well. Optical recording with a voltage-sensitive dye in rat visual cortical slices demonstrated significant suppression in intracortical spread of excitement in the presence of ACH. Some forms of learning and plasticity in cortex appear dependent on the presence of acetylcholine. Baer al. 1986, found that the typical synaptic remapping in striate cortex that occurs during monocular deprivation is reduced when there is a depletion of cholinergic projections to that region of cortex. Kilgard al. 1998, found that repeated stimulation of the basal forebrain, a primary source of ACH neurons, paired with presentation of a tone at a specific frequency, resulted in remapping of the auditory cortex to better suit processing of that tone. Baskerville al. 1996, investigated the role of ACH in experience-dependent plasticity by depleting cholinergic inputs to the barrel cortex of rats. The cholinergic depleted animals had a significantly reduced amount of whisker-pairing plasticity. Apart from the cortical areas, Crespo al. 2006, found that the activation of nicotinic and muscarinic receptors in the nucleus accumbens is necessary for the acquisition of an appetitive task. ACH has been implicated in the reporting of expected uncertainty in the environment based both on the suggested functions listed above and results recorded while subjects perform a behavioral cueing task. Reaction time difference between correctly cued trials and incorrectly cued trials, called the cue validity, was found to vary inversely with ACH levels in primates with pharmacologically, for example Witt al. 1997, and surgically, for example Vutko al. 1994, altered levels of ACH. The result was also found in Alzheimer's disease patients, Parasraman al. 1992, and smokers after nicotine, an ACH agonist, consumption. The inverse covariance is consistent with the interpretation of ACH as representing expected uncertainty in the environment, further supporting this claim.
Synthesis and degradation.
Acetylcholine is synthesized in certain neurons by the enzyme choline acetyltransferase from the compounds choline and acetyl-CoA. Cholinergic neurons are capable of producing ACH. An example of a central cholinergic area is the nucleus basilis of minert in the basal forebrain. The enzyme acetylcholinesterase converts acetylcholine into the inactive metabolites choline and acetate. This enzyme is abundant in the synaptic cleft, and its role in rapidly clearing free acetylcholine from the synapse is essential for proper muscle function. Certain neurotoxins work by inhibiting acetylcholinesterase, thus leading to excess acetylcholine at the neuromuscular junction, causing paralysis of the muscles needed for breathing and stopping the beating of the heart.
Receptors.
There are two main classes of acetylcholine receptor, ACHR, nicotinic acetylcholine receptors, NACHR, and muscarinic acetylcholine receptors, MACHR. They are named for the ligands used to activate the receptors.
Nicotinic.
Nicotinic ACHRs are ionotropic receptors permeable to sodium, potassium, and calcium ions. They are stimulated by nicotine and acetylcholine. They are of two main types, muscle type and neuronal type. The former can be selectively blocked by Cura and the latter by hexamethonium. The main location of nicotinic ACHRs is on muscle end plates, on autonomic ganglia, both sympathetic and parasympathetic, and in the CNS.
Myasthenia gravis.
The disease myasthenia gravis, characterized by muscle weakness and fatigue, occurs when the body inappropriately produces antibodies against acetylcholine nicotinic receptors, and thus inhibits proper acetylcholine signal transmission. Over time, the motor end plate is destroyed. Drugs that competitively inhibit acetylcholinesterase, for example, nostigmin, physostigmin, or primarily pyridostigmin, are effective in treating this disorder. They allow endogenously released acetylcholine more time to interact with its respective receptor before being inactivated by acetylcholinesterase in the synaptic cleft, the space between nerve and muscle.
Muscarinic.
Muscarinic receptors are metabotropic, and affect neurons over a longer time frame. They are stimulated by muscarine and acetylcholine. Muscarinic receptors are found in both the central nervous system and in the peripheral nervous system of the heart, lungs, upper GI tract, and sweat glands. ACH is sometimes used during cataract surgery to produce rapid constriction of the pupil. Atropine extracted from the plant deadly nightshade produces the opposite effect, anticholinergic, by blocking of the muscarinic ACHRS and thereby increasing pupil size, dilation. Now, it must be administered intraocularly because corneal cholinesterase metabolizes topically administered ACH before it can diffuse into the eye. Similar drugs are used to induce mydriasis, dilation of the pupil, in cardiopulmonary resuscitation and many other situations.
Drugs acting on the cholinergic system.
Blocking, hindering or mimicking the action of acetylcholine has many uses in medicine. Drugs acting on the acetylcholine system are either agonists to the receptors, stimulating the system, or antagonists, inhibiting it.
Receptor agonists/antagonists.
Acetylcholine receptor agonists and antagonists can either have an effect directly on the receptors or exert their effects indirectly, for example, by affecting the enzyme acetylcholinesterase, which degrades the receptor ligand. Agonists increase the level of receptor activation, antagonists reduce it.
Associated disorders.
ACH receptor agonists are used to treat myasthenia gravis and Alzheimer's disease.
Alzheimer's disease.
Since A4B2 ACHRS are reduced in Alzheimer's disease, drugs that inhibit acetylcholinesterase, for example galantamine hydrobromide, a competitive and reversible cholinesterase inhibitor, are commonly used in its treatment.
Direct acting.
These are drugs that mimic acetylcholine on the receptor. In low doses, they stimulate the receptors, in high doses they numb them due to depolarization block.
Cholinesterase inhibitors.
Most indirect-acting ACH receptor agonists work by inhibiting the enzyme acetylcholinesterase. The resulting accumulation of acetylcholine causes continuous stimulation of the muscles, glands, and central nervous system. They are examples of enzyme inhibitors, and increase the action of acetylcholine by delaying its degradation. Some have been used as nerve agents, sarin and VX nerve gas, or pesticides, organophosphates and the carbamates. In clinical use, they are administered to reverse the action of muscle relaxants, to treat myasthenia gravis, and to treat symptoms of Alzheimer's disease, rivastigmin, which increases cholinergic activity in the brain.
Reversible.
The following substances reversibly inhibit the enzyme acetylcholinesterase, which breaks down acetylcholine, thereby increasing acetylcholine levels. Many medications in Alzheimer's disease. Donepezil, galantamine, rivastigmin, tacrin, donepezil, galantamine, rivastigmin, tacrin, edrophonium, differs myasthenic and cholinergic crisis, nostigmin, commonly used to reverse the effect of neuromuscular blockers used in anesthesia, or less often in myasthenia gravis, physostigmin, in glaucoma and anticholinergic drug overdoses, pyridostigmin, in myasthenia gravis, carbamate insecticides, for example, aldolab, huperzine A.
Irreversible.
Semi-permanently inhibit the enzyme acetylcholinesterase. Ichathiophate, isofluorophate, organophosphate insecticides, malathion, parathion, azimphos methyl, chlorpyrifos, among others, organophosphate-containing nerve agents, for example, sarin, VX. Victims of organophosphate-containing nerve agents commonly die of suffocation, as they cannot relax their diaphragm.
Reactivation of acetylcholine esterase.
Pralidoxime.
Receptor antagonists. Antimuscarinic agents.
Atropine, ipratropium, scopolamine, sheetopium, diphenhydramine.
Ganglionic blockers.
Mechemelamine, hexamethonium, trimethophan, bupropion, dextromethorphan.
Synthesis inhibitors.
Organic mercurial compounds, such as methylmercury, have a high affinity for sulfhydryl groups, which causes dysfunction of the enzyme choline acetyltransferase. This inhibition may lead to acetylcholine deficiency, and can have consequences on motor function.
Choline reuptake inhibitors.
Hemicholine.
Release inhibitors.
Botulinum acts by suppressing the release of acetylcholine, whereas the venom from a black widow spider, alpha-latrotoxin, has the reverse effect. ACH inhibition causes paralysis. When bitten by a black widow spider, one experiences the wastage of ACH supplies and the muscles begin to contract. If and when the supply is depleted, paralysis occurs.
Other uncategorized unknown.
Surrogate toxin.
Notes.
Campbell, N.A. Rees, J.B. 2002. 48 inches. Biology, 6th ed. San Francisco, California, Pearson Education, Incorporated, pages 1037. ISBN 0-8053-6624-5. Jones, B.E., 2005. From Waking to Sleeping, Neuronal and Chemical Substrates. Trends in Pharmacological Sciences 26, 11, 578-86 doi, 10.1016/J-Tips. 2005.09.009. PMID 16183137, Himmelheber, A.M. Sata, M. Bruno, J.P., 2000. Increases in cortical acetylcholine release during sustained attention performance in rats. Brain Research. Cognitive Brain Research 9, 3, 313-25 doi, 10.1016 per second 0926-6410, 000012-4. PMID 10808142. http://www.biopsychiatry.com/ Alzheimer.htm, Platt, Bettina. Riddle, Gnott, 2011. The cholinergic system, EEG in sleep. Behavioral Brain Research 221, 2, 499-504 doi, 10.1016/JBBR. 2011.01.017. PMID 21238497, http://brafoundation.org/ Discoveries/ Potential-root-cause-of-depression-discovered-by-NASA-grantee, A.B. Wolf, N.J. Butcher, L.L., 1986. Cholinergic systems in the rat brain, 3. Projections from the pontomescent cephalic tegmentum to the thalamus, tectum, basal ganglia, and basal forebrain. Brain Research Bulletin 16, 5, 603-37 doi, 10.1016/0361-9230, 86, 90134-6. PMID 3742247, A.B. Wolf, N.J. Butcher, L.L., 1989. Cholinergic systems in the rat brain, 4. Descending projections of the pontomescent cephalic tegmentum. Brain Research Bulletin 23, 6, 519-40 doi, 10.1016/0361-9230, 89, 90197-4. PMID 2611694, Goldberg, J. A. Reynolds, J. N. J. 2011. Spontaneous firing and evoked pauses in the tonically active cholinergic interneurons of the striatum. Neuroscience 198, 27-43 doi, 10.1016/J Neuroscience. 2011.08.067. PMID 21925242. Edit, Morris, G. Arcadia, D. Nevitt, A. Vordier, E. Bergman, H. 2004. Coincident but distinct messages of midbrain dopamine and striatal tonically active neurons. Neuron 43, 133-143 doi, 10.1016/J neuron. 2004.06.012. PMID 15233923. Edit, Haj-Dalmain, S. Andrade, R. 1996. Muscarinic activation of a voltage-dependent cation non-selective current in rat association cortex. Journal of Neuroscience 16, 12, 3848-61. PMID 8656279, ABC Ekgerman, E. Feldmeier, D. 2009. Cholinergic filtering in the recurrent excitatory microcircuit of cortical layer 4 inches. Proceedings of the National Academy of Sciences of the United States of America 106, 28, 11753-8 doi, 10.1073/FNAUS 0810062106. PMC 2710689. PMID 19564614, College, A.T. Stewart, G.J., 2005. Cholinergic inhibition of neocortical pyramidal neurons. Journal of Neuroscience 25, 44, 10308-20 doi, 10.1523/Shirossi 2697-05.2005. PMID 16267239, College, A.T. Baki, D.J. Zhang, S.S. Matsui, M. Ye, H.H., 2009. M1 receptors mediate cholinergic modulation of excitability in neocortical pyramidal neurons. Journal of Neuroscience 29, 31, 9888-902 doi, 10.1523/Shirossi 1366-09.2009. PMC 2745329. PMID 19657040. Parekh, B. Kozak, R. Martinez, B. Sata, M. 2007. Frontal acetylcholine release controls cue detection on multiple time scales. Neuron 56, 141-54 doi, 10.1016/J neuron. 2007.08.025. PMC 2084212. PMID 17920021, College, A.T. Park, S.B. Kawaguchi, Y. Stewart, G.J., 2007. Heterogeneity of phasic cholinergic signaling in neocortical neurons. Journal of Neurophysiology 97, 3, 2215-29 doi, 10.1152/JN00493.2006. PMID 17122323. Spalmanar, Daniels J.C., Smathers C.C., 1971. Acetylcholine and the synaptic transmission of specific impulses to the visual cortex. Brain 94, 1, 125-38 doi, 10.1093/brain/94.1.125. PMID 4324030, Foot S.L., Friedman R., Oliver A.P., March 1975. Effects of putative neurotransmitters on neuronal activity in monkey auditory cortex. Brain Res 86, 229-42 doi, 10.1016/0006-8993, 75, 90699X. PMID 234774, Stone T.W., September 1972. Cholinergic mechanisms in the rat somatosensory cerebral cortex. J. Feisel. Lond. 225, 485-99. PMC 1331117. PMID 5074408, McGarvey J., Kaiser T., Sartor M., April 1996. Behavioral vigilance following infusions of 192 IgG saporin into the basal forebrain, selectivity of the behavioral impairment and relation to cortical 8-positive fiber density. Buoff. Neurosi. 110, 247-65 doi, 10.1037/0735-7044.110.2.247. PMID 8731052, ABCACY, Cruikshank S.J., Metherate R., October 2000. Differential modulation of auditory thalamocortical and intracortical synaptic transmission by cholinergic agonist. Brain Res 880, 1-2, 51-64 doi, 10.1016 per second 0006-8993, 00-02766-9. PMID 11032989, Abigail Z., Connors B.W., Amitai Y., September 1997. Differential regulation of neocortical synapses by neuromodulators and activity. Neuron 19, 3, 679-86 doi, 10.1016 per second 0896-6273, 80380-3. PMID 9331357, Kimura F., Fukuda M., Tsumoto T., October 1999. Acetylcholine suppresses the spread of excitation in the visual cortex revealed by optical recording, possible differential effect depending on the source of input. J. Neurosi. 11, 10, 3597-609 doi, 10.1046 per joule 1460-9568.1999.00779X. PMID 10564367, Bear M.F., Singer W., 1986. Modulation of visual cortical plasticity by acetylcholine and noradrenaline. Nature 320, 6058, 172-6 doi, 10.1038/320172 AO. PMID 3005879, Kilgard MP, Mezenisham, March 1998. Cortical map reorganization enabled by nucleus basalis activity. Science 279, 5357, 1714-8 doi, 10.1126/Science. 279, 5357, 1714. PMID 9497289, Baskerville K.A., Schweitzer J.B., Heron P., October 1997. Effects of cholinergic depletion on experience-dependent plasticity in the cortex of the rat. Neuroscience 80, 4, 1159-69 doi, 10.1016 per second 0306-4522, 97, 00064X. PMID 9284068, Crespo J.A., Storm K., Saria A., Zoranit G., May 2006. Activation of muscarinic and nicotinic acetylcholine receptors in the nucleus accumbens core is necessary for the acquisition of drug reinforcement. J. Neurossi. 26, 22, 6004-10 doi, 10.1523/Shirossi 4494-05.2006. PMID 16,738,243, U and Down 2005, with EA, Morocco RT, August 1997. Alteration of brain noradrenergic activity in rhesus monkeys affects the alerting component of cover orienting. Psychopharmacology, Burl. 132, 4, 315-23 doi, 10.1007 per second 002130050351. PMID 9298508, Wutko Mill, Alton D.S., Richardson R.T., Gorman Lake, Tobin J.R., Price-Dale, January 1994. Basal forebrain lesions in monkeys disrupt attention but not learning and memory. J. Neurossi. 14, 1, 167-86. PMID 8283232, Paris Romanar, Greenwood PM, Haxby J.V., Grady C.L., June 1992. Visuospatial attention in dementia of the Alzheimer type. Brain 115, PT3, 711-33 doi, 10.1093/brain/115.3.711. PMID 1,628,198, with EA, Davidson M.C., Morocco R.T., August 1997. Effects of altering brain cholinergic activity on cover orienting of attention, comparison of monkey and human performance. Psychopharmacology, Burl. 132, 4, 324-34 doi, 10.1007 per second 002130050352. PMID 9298509, Katsong, B.G., 2003. Basic and Clinical Pharmacology, 9th ed. McGraw-Hill Medical. ISBN 0-07-141092-9, Knowledge, Carr. Mitch, D. Berezowski, V. Sekeli, R., October 2004. Carnitine, Transport and Physiological Functions in the Brain. Molecular Aspects of Medicine 25, 5-6, 551-67 doi, 10.1016/J-MAM. 2004.06.001. ISSN 0098-2997. PMID 15,363,640.